Okay. So like Dr. Carrier said, my name is Jason Molitoris, and we're going to talk a little bit about cancer and molecular signaling. I think that in addition to that, one of the things that I've incorporated into this talk is kind of how I learned this information as we go through the lecture today. And as I went through the lecture when I took this course 6 or 7 years ago, it is very challenging for me to synthesize this information in a way that I can hold on to. And so these are the things that we're going to talk about. But even, I did a PhD in cancer biology and immunology and learned a lot about some of these different pathways through that process. And I still felt like when I sat down to study for these examinations that these topics were alphabet soup. So how did I make this make sense? This is maybe one strategy in which you can use to try to make this lecture and maybe some of the other ones that you guys are talking about make sense in a way that will facilitate examination success. So the ABR puts out guidelines, and they identify the areas that you need to know, and then they'll give you questions. So the way that I did this was I would organize and write out the major pathways that were important for the exam and important for radiobiology. This is the first paper that I wrote, okay? And I quickly realized that the first sheet of paper and my first outline was kind of too small and not going to give me the space to put everything in. So I rewrote everything out again on another sheet and kind of was able to take all of these different aspects of radiation sensitivity, radiation biology, and try to condense them down in a way that makes sense and see where things would fit together with one another. There's also a bunch of questions, practice examinations and study guides that are provided for these examinations. And so I would literally take the question, I would do a set of questions from one of the years of questions in a particular topic. I'd read the answers to those questions, oftentimes missing the vast majority of them. And then I would go to Hall or other text materials and I would read the text. And I found that if I did things in that way, that reading the text was more meaningful, or listening to a lecture was more meaningful, because it gave me a framework for how is the test thinking about this information? How is the test going to ask questions about this information? Can I anticipate areas in which they consistently ask similar questions so that I'm able to answer those in a reasonable fashion? So during the course of this lecture today, we're going to try this out. So here are 3 questions. All right, I'm going to read them out, and you can make a note about what you think the answers are, and then we'll come back to them towards the end of the lecture. So mutations in growth factor receptors are common alterations in cancer. And these mutation and growth factor receptors can signal cells to enter senescence, directly inhibit protein translation, cause formation of gamma-2 AX foci, result in constitutive kinase activity, leading to cell proliferation, or stimulate ubiquitination of caspase-3 and induce apoptosis. So all of these are important pathways, but the question is related to how mutations in growth factors may participate in malignant transformation. Question number 1. Question number 2. RAS functions as so this is a purely alphabet soup question, purely memorization. How does RAS function? Is it a GTPase, a protein kinase, a phosphatidylinositol kinase, a phosphatase, or a transcription factor? And the final question: the phenomenon of oncodine addiction most closely refers to which clinical scenario? Okay, I think that the test is moving more towards clinical applications of cancer biology. So is oncodine addiction a CML patient treated with imatinib? Is it an EGFR mutant lung adenocarcinoma treated with bevacizumab? Is it a BRAF mutant melanoma treated with ipilimumab? Is it a ALK positive lung cancer patient treated with olaparib? Or is it a CML patient treated with interferon? Okay, so you kind of got to know a couple of things. You got to know what that gene product is and what that drug does in order to answer that question. All right, what are the characteristics of tumor cells? The kind of fundamental 6 characteristics of tumor cells are highlighted on this sheet. They have to be self-sufficient. They have to be insensitive to external stimuli. They have to have the ability to invade and or metastasize. They have to be able to replicate without having senescence. They need to be able to recruit blood vessels, and they need to be able to avoid internal signals that would tell them, both I guess internal and external signals, to tell them that they should undergo apoptosis or cell death. So these are the kind of 6 fundamental properties. You could almost kind of, evading apoptosis and kind of goes with the immunotherapy picture, because a lot of the molecular signals from the immune system will engage a apoptosis pathway within a cell. But the other one would be kind of the other thing would be to avoid the immune system. You could almost add that as a 7th. It does fit into that evading apoptosis type of a paradigm, though. So we'll kind of go through some of these in a little bit more specific. Self-sufficiency and growth signals. So example of this is TGF-beta. So TGF-beta can stop the cell cycle in the G1 phase, all right, and induce differentiation or promote apoptosis. So this is an external signal that can kind of tell other cells, hey, stop, slow down, don't grow, or differentiate from a pluripotent cell to a kind of end of line cell that then may not have a lot of the other machinery that's required for malignant transformation. So that's kind of one example of this, but again, not being able to or being able to resist external stimuli is one aspect of this. So we can move on. Evading apoptosis. So apoptosis is it is a controlled cell death process. This is very important in the development pathways associated with embryogenesis, and so also very important throughout the lifespan of an organism, and so something that commonly happens and something that the cells need to be able to avoid. Most common method for loss of apoptosis is through the p53 protein, and more than half of cancers have a p53 mutation in them, resulting in either a missing or non-functional or depressed function of p53. We'll talk about that a little bit more later. Cancer cells can also produce excessive amounts of anti-apoptotic proteins. So there's this whole BCL2 family of proteins that you need to at least be familiar with. And the BCL2 family of proteins has anti-apoptotic and pro-apoptotic proteins. So if a cell is able to increase the expression of the anti-apoptotic proteins like BCL2, then they're able to inhibit the ability of apoptotic signals, either external or internal stimuli, downstream, to avoid basically mitochondrial destruction of the cell. Sustained angiogenesis. So this is one of those areas that in the last, maybe in the 90s and early 2000s was one of the hottest areas in cancer research. And the concept that cancer cells release molecules, release signals to create new blood vessels, all right? And that makes sense, that they would require increased vascularity as tumors get bigger. We all appreciate the idea of large tumors with necrotic cores, but those tumors still are producing these factors to try and allow for continued growth. So how is angiogenesis regulated? There's a counterbalance of these positive and negative signals. And when there's a tumor, it learns how to secrete these factors like VEGF to take that balance out of proportion and allow for increased blood flow. Cancer cells can also stop producing angiogenesis inhibitors, like thrombospondin 1, which is a potent inhibitor, again, under the control of p53. So again, another way in which p53 mitigates cancer progression in a number of mechanisms. The story about like bevacizumab. So bevacizumab, Avastin, is a monoclonal antibody, one of the first monoclonal antibodies used to target cancers. We have a lot more of those now, but the thought was that if we give bevacizumab to somebody with a cancer, that we will repress the VEGF signal, okay, and we'll be able to stop blood growth, stop the angiogenesis phenomenon around tumors. We'll be able to either kill or decrease growth of these tumors. It was given. They had positive results in a number of cancers. We still use bevacizumab to this day and other anti-angiogenic medications. But there's been, fast forward 5, 10, 15 years, there was actually an increasing amount of information that the bevacizumab was doing almost the opposite thing of what we were thinking. The angiogenesis was so dysregulated within some of these tumors that administration of bevacizumab was actually increasing blood flow or increasing functional blood flow to tumors and allowing for other treatments like chemotherapeutics to actually penetrate the tumors better. And so even in situations where we kind of think we're doing the one thing, we could actually have the opposite effect, opposite mechanism, but the same outcome. So an improved response to overall systemic therapy. Next aspect of this is tissue invasion and metastasis. So this has to do a lot with matrigel proteins that are on the outside of the cell surface. So E-cadherin is a calcium-dependent cell cycle adhesion glycoprotein, and that is loss of E-cadherin function allows for these cells to become a little bit more slippery, whether that's local slippery or getting into the bloodstream and moving through the bloodstream to metastasize to a distant organ. Okay? Other aspect that is interesting about these hallmarks of cancer is that they can kind of occur at a different different pathways. So you can have sustained growth signals, and then you can have removal of apoptosis, and you can kind of work your way through these hallmarks of cancer in different ways. Again, you do have to end up at a place where you have all of these in order to be a malignancy. But it is interesting to think about these pathways, these changes occurring in a differential fashion. And I think that even within, if you take this down to even within a solitary cancer, in a one person's malignancy, just because they have we diagnose them with one cancer, you have a prostate cancer or a head and neck cancer, they have clones of cells that are going through these processes slightly differently. And so you can end up with different rearrangements in different aspects of tumors. And so that's something that, you know, we're now getting to the point where we can do genetics on an individual tumor. Down the road, are we going to be in a place where we're doing genetics on different clones of tumors, and how will that impact things? Okay. All right, so the malignant transformation results from mutations in 3 types of genes. So it's really important to understand these groups of genes, and as we go through the different pathways, you will hopefully we'll be able to talk about some of these aspects again. So gain of function mutations are when a gene becomes more active, and that occurs if you think about this from the point of view of the cancer, from like an anthropomorphic standpoint, the cancer wants to activate genes, oncogenes, or activate genes that cause it to grow. Okay, and then it wants to inactivate tumor suppressor genes, or genes that would either cause it to stop growing or die. And then there needs to be a change in the DNA stability genes. So we need to be able to continue to replicate and divide even in the setting of damaged DNA. One of the biggest signals in a normal cell to stop dividing is damaged DNA. Okay, and so the ability to get rid of those signals is going to be critical for these malignancies. All right, and so we'll kind of talk about a couple of these groups. We'll talk about oncogenes, then tumor suppressor genes, and then we'll get to a couple of smaller areas. So oncogenes, there are a number of oncogenes. This is probably oncogenes and tumor suppressors are kind of the first break point. And when in the 80s and 90s, people were trying to clone these genes out, identify them, identify mutations in genes that led to malignancies. And so here's a list of oncogenes, as well as kind of ways in which oncogenes become activated. So it's kind of important to know the oncogenes themselves, but also to understand the concepts of how a gene that's normally in our body, normally expressed in our body, goes from something that is part of a normal cell life to something that is influencing steps towards malignancy. And so there can just be increased expression. So EGFR, the EGFR receptor on the cell surface, is a good example of that. There can be alterations in the proteins. So RAS, HRAS, NRAS, KRAS, all of these are can have alterations in their DNA, changes in the protein structure, and increased activation of those cells. You can have insertions. So those are a little bit, I think, less tested on, but you theoretically, right, can have an insertion within a gene. You could have a deletion in a gene. NF-kappa B is a good example of a deletion that leads to activation of an oncogene. And then you can have translocations. So translocations are some of the things that are most well known, because those translocations are things that we have been able to intervene on. So like the ABL protein in the Philadelphia chromosome. You can also have increased expression of the anti-apoptotic oncogene BCL2 or MYC through a translocation of these proteins. Okay, the MYC and BCL2 can be part of lymphomas pretty commonly. Okay. You can have changes in signal transduction and growth factors. So, there's a constant amount of growth factor stimulation within the bloodstream, and so you could have overexpression of growth factors that leads to maybe signaling, kind of catching those external signals more that allow for growth factor transformation within the nucleus. But you can also have overexpression of some of these receptors and again, this is a scenario where we thought, okay, well, the EGFR receptor is overexpressed and there's more signal, and so then that's how it happens. But actually, you can also have overexpression of some of these receptors, and then just by virtue of the fact that they're overexpressed, they can kind of signal amongst themselves even without an external factor, to lead to behaving like an oncogene. So sometimes it's not even that these receptors are getting a signal from an external source. They're just kind of enough of them on the cell surface membrane that they're able to, when they kind of bump into each other, they activate. These lead to downstream transcription factors like MYC, Jun, and FOS, through multiple different pathways. We have some examples of those pathways later on, in the talk. Here's an example of that EGFR in the signaling cascade, and constitutive activation through the mutation. Right. This is a long list of proteins, but demonstrates a lot of work that has been done to characterize how EGFR signals within a cell. Okay, it happens through the PI3 kinase P10 AKT pathway. This activation leads. This is a kinase pathway. Okay, and it leads to cell survival and progression, as well as activation of the cellular metabolism and kind of preservation of metabolism, even in spite of limited nutrient and a limited nutrient environment. And then this also will act through the MAP and ERK pathway, as a way to signal for cell growth. There are a number of other genes in here that are critical, and we'll talk about a little bit later, that can be downstream of EGFR. So even if EGFR expression is, let's say, normal, you can still activate these pathways. Okay, that can be done. P10 is a good example of a pathway that can be modified, and then you could have downstream, essentially release of the AKT signaling, or you could have mutant RAS, which we saw on a couple of slides ago with point mutations, and then you could have kind of constitutive activation of RAS, and so you could be downstream of the EGFR receptor, but still generating a signal almost as if the EGFR receptor was being activated. All right, so here's a graph showing the number of tumors that overexpress EGFR. All right, and so if you were to look at this list, you might say, could we, which cancer or cancers would you potentially target with anti-EGFR therapies? Okay, there's only one of these here with 100. And so cetuximab, which is a monoclonal antibody targeting EGFR, was tried in head and neck cancer. While the initial results were relatively positive, I imagine everybody that is in a radiation oncology residency now knows that some of the head-to-head trials with cisplatin were less positive than previous trials had indicated. So, but again, those are mechanisms of activation within head and neck cancer, and then efforts on our half, on behalf of our patients to try and improve care by going after those pathways. Okay, just like we talked about 2 slides ago, RAS is downstream of the EGFR receptor, among other things, and RAS is it's called a GTPase type of a protein, and it's important to understand these mechanisms. So RAS has a GDP attached to it in its inactive state, and then upstream signaling pathways through EGFR or other ways can convert it into a active GTP mediated RAS function. This leads to downstream signaling and cell growth. That GTP is inactivated back into the GDP form by another protein. Okay, that that transition back into the inactive form can be blocked. RAS can be mutated such that it always looks like it has a GTP to activate signaling downstream. Here's a good example of the RAS pathway, kind of just for reference to get an idea of downstream. So downstream, it will activate the MAP kinase pathway, MEK and ERK, leading to gene transcription within the nucleus. Again, here are a number of malignancies with the percentages of RAS mutation. MYC is probably one of the other most commonly tested on and most commonly talked about oncogenes. It's one of the earlier ones that was identified. And 70% of cancers have a deregulated MYC. Chromosomal translocations increase MYC expression. So Burkitt's lymphoma and some other lymphoid malignancies are do this. This is part of that, single hit, double hit that we talk sometimes about in radiation oncology with respect to lymphomas. So MYC is one of those genes. The other one is BCL2. Double hit lymphomas tend to have a poorer prognosis because you have you've increased expression of an oncogene and decreased and you've increased a pro-growth signal with MYC, and you have increased the anti-death signal with BCL2. And so you're kind of hitting both sides of that growth and death equation when both of those are impacted. It can have gene amplification in AML, lung, breast cancer, colon cancer, brain, and prostate, and it can have point mutations that increase its activation function. So again, a number of different ways in which MYC is activated in malignancies, and a very important driver of cell growth, a very important oncogene. Here's an example of translocations in MYC. Burkitt's lymphoma is probably the most kind of well characterized and outlined here. Makes it more transcriptionally active, increases expression levels. And in contrast to the BCR-ABL Philadelphia chromosome, MYC translocations do not alter the protein structure. They largely increase its expression level of the wild type gene and protein. So it's really in MYC, even the translocations are largely impacting the transcription of the MYC protein, whereas in the BCR-ABL, that BCR-ABL complex in the Philadelphia chromosome leads to a constitutively active ABL format, which is kind of different mechanisms and something that they could potentially test on. This is an example here of how MYC functions within the cell cycle. So the cell cycle is a little bit outside the scope of this lecture, but I know it's obviously part of the course and part of the exam. But expression in MYC induces activation of cyclin D1, CDK4, okay, and then CDK6 complexes. That's in the G0 to S transition, or G1 to S transition. And downstream events, cyclin E, cyclin D, CDK2, and cyclin A, CDK2 complexes antagonize association with P15, P27, and P21. So P15, P21, and P27 can block the cell cycle prior to the S phase. MYC will block those proteins, thereby kind of allowing for the cell cycle to go. Okay, and then when I was making my sheet, all of this stuff ends up being built into the pathways as I was iteratively going through the different aspects of this. So we're talking about molecular signaling and oncogenes and tumor suppressors, but that doesn't mean that it's not related to the cell cycle and all of the important aspects of cell cycle regulation. Here's a slide on the Philadelphia chromosome. which is, I think, well known to a lot of people in our field just due to the novelty of the therapeutic targeting of this, as well as the ability to identify it as a potential therapeutic target. So you can see here that downstream of the BCR-ABL protein, we'll have PI3 kinase and AKT pathway, which we've already discussed briefly, the RAS pathway, which we've already discussed as well. And then going into the cell cycle through the STAT3 and STAT5 proteins. Imatinib or Gleevec is the active agent that inhibits ABL by binding to the ATP binding site within the kinase domain. And it was, kind of one of the, I think it was the first targeted therapy that we had to an oncogene like this. And so very, very famous. This tyrosine kinase inhibitor class of medicines has obviously grown dramatically over the last 20 years. Okay, so we've gone from imatinib to dasatinib to now we've got when I go to GI tumor board, every second drug out of the mouths of the medical oncologists ends in IB. So regorafenib, there's all kinds of these medications that are used, colorectal cancer, renal cancers, along with the lymphomas where they were kind of initially developed. So very, very important class of medicines. Again, these are all kind of inhibiting these tyrosine kinases, just like Gleevec and the ABL protein. So oncogenes do induce radioresistance. So this is kind of, if you look at model systems where you increase oncogene expression inside of cells, you will actually induce radioresistance. So you get less survival with the BCR-ABL, MYC, or RAS in comparison to the wild type cells here. Again, so things that that gives us the sense that, well, maybe we should be trying to target these as in cetuximab or other things while we're giving radiation. Okay, the other big class of genes other than the oncogenes is the tumor suppressor genes. Okay, so these are the breaks. These are the proteins. These are the molecules inside a cell that say, whoa, like let's you guys are kind of going out of control here. Let's slow everything down. So P10, which we've seen on some previous slides, P53, which we've talked about, the repair genes, so genes that will identify and signal within a cell that there's DNA damage and stop the cell from being able to progress, like the BRCA genes. And then there's cell cycle inhibitors, retinoblastoma, and then apoptosis inducers. So there's multiple members of the BCL2 family that induce apoptosis. So BCL2, again, BCL2 prevents apoptosis, but there are other members of the family that work in opposition to BCL2. That's BAX, BAD, the BIM, the BH3 only proteins. These decrease. So if you remove a tumor, if you have loss of function in these, then you have a lack of cell cycle arrest. So these cause cell cycle arrest. You remove it, kind of, I think about it like a double negative. Then you get lack of cell cycle arrest, and you get decreased cell death, and you get you can have increased ability to metastasize. Okay, again, these fall onto the sheet. So retinoblastoma was really the first tumor suppressor that was identified. This is from the Knudsen and the kind of understanding of genetics with respect to retinoblastoma in kids. So there's kind of hereditary and non-hereditary retinoblastoma that were identified, and the question was like, why why is this the case? And so the hereditary forms are kids that have one functional gene of retinoblastoma and one non-functional. Okay, so they have, instead of having two brake pedals ready to go, there's only one. And so as soon as there's a stimulus or as soon as there's that other brake gets removed, then you can go right into having retinoblastoma. The peak of incidence is younger, can happen in both eyes, and obviously has implications other than just retinoblastoma. It can have other types of malignancies, because now you've already got rid of, you're already working on some of those 6 characteristics of cancer, right? So that's hereditary. Non-hereditary is in a person with normal retinoblastoma on both brakes. So they have both brakes, but there's still a chance that both of those get pulled. This peaks a little bit later in life. It's very unlikely to affect both eyes because it's unlikely to have one hit, let alone 2 hits. And then secondary cancers is going to be much lower as well. So here's just some pictures of retinoblastoma. And then the 2 ways that this happens: hereditary on the left, and then sporadic on the right. So if you have somebody who has let's say an early retinoblastoma, or has genetics done, and they know that they've only got one active function of the RB protein, there's obviously a 50-50 chance they pass on the inactive form to their offspring, in which case they would be at that higher risk for development of retinoblastoma and other malignancies. Second allele of retinoblastoma can be lost by a number of mechanisms. Okay, so again, we talked about the oncogenes and ways that those could be changed in such a way that they become more of a gas pedal, right? So they could have a point mutation that causes them to be constitutively active. They could be just overexpressed. They could have a translocation like MYC that leads to increased expression of the protein. They could have translocation so that they're bound to something else, such that they now have a constitutive active form. So same thing with these tumor suppressor genes. They could have a point mutation, they could have non-disjunction, they could have recombination, all of which could lead to the protein becoming non-functional. Okay, it's I think it kind of makes a little bit more sense to me to take something that's functional and make it non-functional, as opposed to something that was either non-functional or partially functional and then increases activation. So this makes sense. There's a number of different ways you could take something, a functioning protein, and get rid of it. All of them would apply to a lot of these different things. So you can have another thing that can happen, especially with these tumor suppressors, is you can have altered chromatin. So again, we think about the genetics, and the genetics are the first thing that we were able to really characterize, because we figured out how to do sequencing on those huge gels, and you could do sequencing, find mutations in genes, mRNA sequences, and create all kinds of cDNA libraries, all this stuff. But then it became very apparent that it's not just the genes, it's also the surrounding structure of the DNA, and so chromatin formation. So tumor suppressors could have their chromatin changed in such a way that the gene is bound up and not accessible to transcription factors and not able to be transcribed. Okay, so there could be no changes in the gene, but the changes in the chromatin really decrease or abolish the expression of the tumor suppressor gene. Okay, so that's depicted here, where they're kind of locked with methylated DNA. Okay, and so good examples of things that we appreciate from a methylation standpoint would be MGMT and in GBM, and as a kind of a good example of this, and somewhere where we have therapeutic targets. All right, and so here's just closed chromatin leading to decreased tumor suppressor formation. All right. Again, you could have a number of you could have kind of, because you have two tumor suppressor proteins, you could have different impacts, different things impact both of them, because all you need to do is get rid of it. Okay. So you can get rid of it by methylation, which is depicted over here on the right-hand side of the screen, and condensing of the chromatin. You could have loss of heterozygosity. You could have a number of different ways in which this could occur. All right, so here's the RB pathway, again, functioning with a peripheral to the cell cycle. All right, the multi-step process here, we talked a little bit about this at the beginning, and so in the interest of time, I'll kind of skip through this, but you can have multiple clones of cancer cells, and you can have different alterations, and colorectal cancer is kind of the best example of this stepwise progression. Okay, and so again, this is something that was relatively straightforward to study compared to other things, and so you could kind of think about, okay, well, you have one change that leads to something being able to grow, and then you have another change that leads to increased vascularization, and then you have another change that leads to the ability to metastasize. And so you can people have characterized these over the course of time with respect to colorectal cancers, and there are some very stepwise functions that happen. The APC mediated familial colorectal cancer is a good example, because you're basically setting up the opportunity to have one thing happen followed by multiple things after that. It's not always how it works, though. There can be obviously very, very significant dramatic changes that occur that would lead to multiple aspects of this occurring instead of a very stepwise function. Same thing with GBMs, and that's kind of outlined here. Right. Caretakers and gatekeepers are kind of important concepts for us to understand. So gatekeepers, they encode gene products that act to prevent growth and potential cancer cells accumulation of mutations. Okay, so these are retinoblastoma, P53, and WNT1. So these really are gatekeepers in that they identify and allow for repair of DNA, so that you don't have accumulation of these of small amounts of mutations. Okay, caretakers encode products that stabilize the genome. All right, so these are ones that just kind of they limit the likelihood that you're going to have mutations. Right, so what you want to do is you want to limit the chance that you get a mutation with the caretakers, and then gatekeepers are like, oh, well, there is a mutation, so let's slow everything down, allow ourselves to fix this, and we'll move forward. Okay. So you can have changes in the DNA damage response, all right, and impact on all of these aspects at the bottom. So when we do DNA damage, obviously that has sensors at the top level, transducers, and then effectors that will block cell cycle transitions, induce apoptosis, and can change transcription and DNA repair. All things that we need to have a good appreciation for. The ATM pathway is obviously critical to early signaling within radiation-induced damage. I mean, that's outlined here. So ATM is obviously something that is important in radiation due to sensitivity for radiation in ATM phenotypes. ATM phosphorylates a number of proteins involved in cell cycle, and cells from the individuals have radiation hypersensitivity. which is and on the thing. P53 is going to be critical for the examinations. It's a transcription factor. It's degraded by binding to MDM2. It's mutated in over 50% of cancers. It is a tumor suppressor, and it is activated by radiation through ATM, the ATM pathway. So this does, when it's activated, it becomes a tumor suppressor. So it leads to things that decrease cancer. It is often mutated, and so those malignancies tend to be less radio responsive. Okay, so things that we can look at as opportunities for increasing the effectiveness of radiation. These are all the ways that P53 works in a radiation-induced system. Okay, a little bit of in vivo data. Here's a bunch of the oncogenes. We've kind of gone through the majority of these, but radiation can lead to EGFR clustering and radio resistance. It can lead to activation of EGFR through the TGF alpha pathway and lead to radio resistance. Okay, this is the data for cetuximab that we talked about in the preclinical model. And then you could potentially other ways that you could inhibit EGFR activation with gold particles. Just concepts for how do we increase radio sensitization and the cancer signaling effects from radiation here. So I'll let you guys read. I want to get to the questions. Here's a quick summary that you guys can take a look at. So let's take a look at our questions. So we've got a few minutes left. And so first question, mutations in growth factor receptors are common alterations in cancer. So what can they do? Can they signal cells to enter senescence? So that senescence would be the cell is kind of inert. So that's not a factor that is going to be kind of pro-oncogenic. Inhibit protein translation. Again, maybe not the most pro-oncogenic thing. Formation of gamma H2AX, foci and cell nuclei, something that happens with radiation, and DNA damage. And then constitutive kinase activity to signal cells to proliferate. That sounds promising. Or stimulate ubiquitination of caspase-3 to induce apoptosis. Again, if we induce apoptosis, we're not pro-oncogenic. So I would vote D based off of what we've talked about today, and that is the case. Let's see. RAS is functions as what? A GTPase, a protein kinase, PI3 kinase, a phosphatase, or a transcription factor. So we talked about RAS. Again, this is just the rote memorization question, but it is RAS is a GTPase. Okay, so these are sometimes the questions that you'll get. And then the phenomenon of oncogene addiction most correctly refers to which of the following clinical scenarios? So a CML patient treated with imatinib, an EGFR mutant lung adenocarcinoma treated with Bev, a BRAF mutant melanoma treated with IPI, ALK positive lung cancer treated with olaparib, or a CML patient treated with interferon. So this one I think is a little bit more tricky. Bevacizumab is an anti-angiogenic. Ipilimumab is an immunologic medication, and interferon is not it, but so it's imatinib. So really it's BCR-ABL is kind of an oncogene addiction. Okay. All right. I'm gonna stop share. Happy to take any questions. Looks like
Thank you, Dr. Molitoris. Do we have any more questions for Dr. Molitoris? I'm not seeing anything in the chat. coming. So thank you so much for taking time from your busy schedule, preparing the lecture and getting out of the clinic. We really appreciate it. And like I mentioned to people before, all the lectures are recorded and they can send us their questions before, and all speakers will be available to answer them at a later time. Thank you.
Thank you.